Hello guys and welcome to it. This is Learn Extra Live on a Wednesday where we're learning more and learning extra on life sciences. My name is Abram and of course it is another day with Cheryl. It is a CA day. Welcome back.
Thank
How
you.
are you?
No, I'm well. Had my break last week. Now ready to go again.
So you were running away from my ear.
Yeah, no.
You didn't want to listen to
you
us.
know what? Hey, you were like, I was just, you were calling me all these funny little abbreviations. So I thought I was so old I had to go for a rest.
I'm so sorry. But welcome back. Welcome back. What do you have for us?
Hi, grade 12s. Tonight we're going to be looking at meiosis. Okay, are you going to carry on? Are you going to do your little thing?
Yeah, I'm going to do my little thing. I thought you were going to explain more. Hey,
nah,
okay,
you know what?
It's
one of those things, just a teaser. Otherwise, if I start explaining, we're not going to stop.
We're not going to stop.
Yeah.
Well, guys, well, I'm so excited for the show. All you need to do is to get your notes. We've got this awesome workbook. You can get it by simply sending an email to sales@mindset.co.za. It has some solutions on it as well. So guys, get it right now. It's only 139 Rand. So ask for that 139 Rand from your parents, or you can just save up to buy yourself this. It's coming very handy, especially now that we're going to be writing our exams soon, metrics, so you need to get it. Otherwise, I've already posted the notes for you on learn.mindset.co.za. You can get them with the schedule, and you can even get the previous shows as well, see what we've been doing. But for today, I have all of them on our Facebook page, which is facebook.com/Learn Extra. Always like our page and tell your friends to also like our page, and let's learn together. On Twitter, we are at Learn Extra. So let's follow each other and talk more.
Sure, you do that so well. Welcome, grade 12s. You will notice we're going to do meiosis tonight. I know most of you did it at the beginning of the year, right? But we didn't do the live shows there, so we decided that what we're going to do is we're going to do a lesson on meiosis. Please remember, when we look at meiosis, it is in paper 1 and it is in paper 2, right? Because we're going to look at it when we do, obviously, meiosis, and we look at it reproduction, and we're going to just deal a little bit about it when we do the evolution. So when you look at the meiosis section, understand and be able to link it to more than one section. Okay, so when we talk about meiosis, what are we talking about? We're talking about the division of a cell. Now, if you can see behind me on the board here, I'm going to take a step back. And I say you need to remember back, and most of you can't. When we look at cell division, we see that there are two different types. And in grade 10, you would have looked, when you did the cell, you would have looked at this process called mitosis first. Okay, when we look at the process mitosis, there is a lot of difference between mitosis and meiosis. So you need to be able to differentiate and distinguish between the two. Let's quickly just recap what mitosis was. If you can remember, one of the things when we're looking at, mitosis is the division of a cell. And when we talk about a division of the cell, we're talking about one cell dividing into two. Now, the key word that I want you to remember here is that mitosis occurs in a body cell. Because when we look at meiosis, we are very particular about where this process occurs. The second thing, I'm going to put a number 1 over here. The second thing that is most important is that at the end of my mitotic division, I have got two cells that are identical. All right? And how did I get that? Let's go back to the beginning of the year when I looked at a process called DNA replication. Okay, so we can already link mitosis back to the DNA that we did at the beginning of the year. So if I were to look at my diagram, here is my one cell, all right, which is my parent cell. Now when we talk about, I'm going to talk about this thing called ploidy. What do we mean by ploidy? And ploidy means that we give a letter, and we use N in this case, to give us a number of a chromosome. Now, what do we mean is when we talk about a somatic cell we refer to as 2N. What does 2N mean? That means it's diploid. All right, why do we use letters? Because not all organisms have the same amount of chromosomes. So when we use N And when we use 2n, we are talking about half the amount, all right? And there we go, is technically what we would call the full amount. So how do we get to 2n? Remember sexual reproduction. We've got a mom, we have a dad or a male and a female, and then we get the zygote, which is 2n, and from that, that's where the cells are going to develop. So, two key things. One thing, 2n. All right? At the very end, after I have got this whole process, and we'll go through the words again, interphase, prophase, metaphase, anaphase, telophase, but at the end, this is what you need to know. How many cells do I have? Let's nerd up a little bit. How many cells do I have? I have 2. And look at the colors. Try and look at why we use colors here. Okay, exactly the same DNA. My 2 cells that I make are identical. So why would I need, all right, for mitosis to take place? We looked at it before when it's almost as similar to why DNA replication needs to take place. To grow, all right, to repair, to replace. So if something in our body is damaged, we can replace it with the same cell. All right, in certain organisms, all right, what do we find? Asexual reproduction. All right, they use the simple process to be able to make new, all right, organisms. Now we need to get to the process of meiosis. Okay. Now, when we look at meiosis, you need to have in the back of your mind this, all right, what did mitosis was. Mitosis, I had 1 cell. What is the end result? I have 2. And what do those 2 cells look like? They are identical, right? You've often heard the word cloning. That is basic, that's what mitosis is. It's cloning, making an identical copy. Now, when it gets to meiosis, that is the one thing we do not want. The one term that you're going to hear very often, all right, when I use meiosis, is this word genetic variation. And I'm sure your teacher has used it a lot of times before, and we're going to use it when we look at evolution. There's this term biodiversity. What do we mean? If I ask you to list 10 animals, you're able to list them, or you can quickly just think about it and list it. What makes them different? And if you're sitting next to your mom or your dad on the couch or your little brother's running around, have a look at them, all right? They look similar to you, but they don't look exactly like you, all right? Because what do we want? We talk about healthy, if we talk about healthy genes, all right, if I can talk about that, we're looking at an individual where there's lots of variety. There's a lot of, we change. We take the little genes and we mix them up and we make something new. And that is the key to meiosis. Right, so let's start. We're going to start meiosis. Always have like a tick point. I always say, what is your end result going to be? And when we look at meiosis, all right, before I show you where it happens, you need to understand, let's have a look at the picture. The first thing you should notice is that at the end, I don't make 2 cells, but the possibility of making 4. All right? If we link this back to reproduction, we know that in the female, only 1 cell, all right, that's the egg cell is going to survive. But we know in the male, the sperm, all right, what do we really want? We want lots and lots and lots of them. Okay, so the first thing that I notice, I have 1, all right, and at the end, all of a sudden I have 4. Okay, so I start off with 1 and I end off with 4. Different to mitosis. If I look at the diagram, what do I notice? I notice that my chromosomes have got all of these different colors. And when I look at the end, I notice that this one's got a little bit of green with orange, this one's got a lot of green, a little bit of orange, and what I notice is each of these cells is different. All right? So, at the end, I started off with 1 cell, and at the end, what did I make? I've got 4. And unlike in mitosis, what happened is, I'm not quite sure, I'm going to explain it now, they all landed up being different. So the DNA in all of them was different. Okay, and that means we have a genetic, let me just move it up a bit, genetic variety or variations. Okay, now if I have a look, at my cell, I look at the chromosomes and I see, okay, I've got 1 chromosome, I've got 2, I've got 3, I've got 4. Now, if I look at the end and I go to the end, what do I notice? Oh dear, in the end, right, what have I got? I've only got 2 cells. So if I started off with 2n, and here my 2n was 4, and here at the end, I ended up with 2, what have I done? I have halved my chromosome number. And now I have a cell that I refer to as haploid. Why do I need to do that? Okay, why do I need to ensure that my chromosome number stays the same? Right, I need to ensure it because that if my chromosome number changes, that might mean that my individual is no longer fertile. And if it's no longer fertile, it means it can't reproduce. And what does an organism need to be able to do to be successful? It needs to make more copies of itself. Okay, so when you look at the meiosis, look at why, all right, does it happen? What do we need to do? And that leads us back to where it happens. So if we go back to where meiosis happens, we are looking at the sex organs. Why? Because the sex, let's change color quickly. Why the sex organs? Because the sex organs in the male's testes and the female's ovaries, what am I going to produce? Here I'm going to produce a sperm, and there, you know me always with the egg, and there is the egg. And those two, all right, what do we need to do? Those two are half of one individual. They are 2 cells that need to join together to become a new organism. So why do I need to have half? Because I need to make half of one person. The male is going to give me half of his DNA. The female is going to give me half of hers. And in doing so, because these cells are the only ones that can fertilize, what are they going to make? They're going to make this whole individual that we call a baby. It's going to happen exactly in plants. In plants, we have reproductive structures. Here's the male. What happens in there? That's the anther. What do I make? Pollen. Right? And when the female, what do I make over here? My ovule is my egg cell. Again, half a cell, so to speak. What are they going to do? They are going to fertilize, and when they fertilize, they're going to make the seed. And that seed will then become, right, a new organism. So what we need to do is we need to keep the same number. But now you need to keep in mind, because we're going to look at the different phases just now, what I need to do is, how do I keep the same number of chromosomes in an individual, yet at the same time, right, I am going to need to bring about the so-called thing called genetic variation. Okay, I know we're almost time for a break, but I want to show you quickly. So when we go on to, right, when we look at meiosis, meiosis is going to consist of 2 divisions. Where mitosis was quite simple, 1. Okay, so what do we have to do? when we look at meiosis, we need to do two things. One, I need to halve my chromosome number, and two, I need to maintain or get this genetic diversity. So what we're going to do in the first one, what am I going to do? I'm going to get that half number, and I'm going to change things around, and then when I've done that, I'm going to be in the process, right, of making 4 cells. Time for a break.
Yes,
and then
of
we'll
course.
come back.
Yes, and thank you guys for tuning in and for all your comments. I know our regular guys that always are tuning in. Shafi Solomon's a big shout out to you. His gift in Chabileng, Refulo Maleka as well as Butsilo. I'm not sure if you're late today or not, but guys, let's keep the conversations going on our Facebook page. See you after this break. Welcome back, guys. Now, if you're studying with us and you don't have the notes, simply jump into our Facebook page, which is facebook.com/learnextra. I've already posted the link for you guys. And I see your questions and your comments, but I need to tell you more about the competition, the South Point Accommodation Competition. All you need to do to enter is to simply fill in the entry form, and that's it, guys. You could win yourself for next year 1 year of free accommodation. Remember, guys, this is only available to you guys, the matriculants. That's how special you are to us. Now, Cheryl, I've got this comment from Refulo saying, AC in the pack, lovely. So he just decided to change our arrangement because we were a CA, so now we are AC. I'm not sure if he's doing physical sciences or not, but I know
fine.
he's doing physical.
If I'm after you, it means I'm younger than you, so I'm fine. Tell him, that's fine. I'm fine.
Well,
yes.
this is his message to you. I just love her and her teaching. I master life sciences. Please, AP, say thanks to her. I just did refill.
Thank you. Thank you so much. Can I carry on?
Yes, you can.
Okay, guys, now we're going to get to the phases. This is the most important thing. If you go through, and if I don't get to all of the questions tonight, please go over the notes. It's very important that you are able to identify a specific phase in each of the 2 divisions. And now in meiosis, it gets very difficult because you don't only have meiosis 1, but you have to meiosis 2. So you need to pay careful attention as to which of the divisions, and I'm going to give you hints so that you know which one we're going to look at. Okay, so now we're going to start with meiosis 1. Now, when we did mitosis, I use an acronym, all right? I know it maybe sounds funny, but I tell my kids to remember something called IPMAT, all right? And as you can see, what IPMAT is going to be, when we look at the phases, we see they happen. There's interphase for the I, there's prophase, there's metaphase, there's anaphase, and there's telophase. So we're going to look at each of the phases. So let's start. When we look at meiosis 1, look at another name for it. Reduction division. Okay, sounds like a very long thing. All that I need to know is, if I start out, no matter what my chromosome number, at the end of this division, all right, I must have half of what I started off with. Okay, so to say 2n, at the end, I must have n plus n. So say, for example, let's take, if I have 20 chromosomes, I'm a special little fly, and I have 20. At the end of meiosis 1, one cell must have 10, the other cell must have 10. Okay, so we're going to be left with half the number of chromosomes. That is the most, I won't say most important, but that is at your end result, that's what you're going to see. Also what you're going to see, right, is that at the end, my chromosomes are going to look different from what they started off. Okay, and that is genetic variation. So let's have a look to see. Now, when it comes to the meiosis, we very often don't talk about interphase, because interphase is a phase that you can't actually see anything happening. So the cell looks like it would do when it's at rest. Okay, but now what is actually happening in the cell is DNA replication. Okay, I'm going to just do a quick little math thing. I just want you to, all right, So now when we do DNA replication, now I want you to imagine, let's look at the human cell. The human cell has got 46 chromosomes in it. At the end of meiosis, I want to have 4 cells with half the number. So what is half of 46? 23. So what is 23 times 4? 46 plus 46, isn't it? So what I need to do is, when I make DNA replication, what it's saying to me is, I'm making enough DNA here so that I can split it between 4, not between 2, like in mitosis. So I can't just make up DNA and put it in a cell. I need to make enough DNA that I can share. Don't you want to share equally? That is what we want to do. Okay, so in interphase, my DNA makes a copy of it, and that technically, all right, those are my cells, and I'm going to share those among the 4. But now, how do I do that? Now, when it comes to meiosis, there's very particular phases that you need to pay attention to. The first one is prophase 1. Now, if I have a look at that cell, and it becomes this cell, what do I notice? Okay, what have I noticed from here to here? I have noticed that during prophase, what has happened? Let me change color. My centrioles have developed, and what develops between them? Spindle fiber. But the most important thing during prophase 1, I see these chromosomes lining up together. And what do I mean together? I mean, the term that you need to learn now is homologous chromosome. What does homologous chromosome mean? If you have a look at your karyotype, remember you've got 1 to 23. This blue one, for argument's sake, is daddy's number 1. That's daddy's. The pink one is mommy's. And the information that is on this chromosome is exactly the same information on that one. So if this little point says eye color, then this little point says eye color. So that is why we say homologous. What does homologous mean? The same. So now we line up, we take all our chromosomes and we line up, we put them into groups. They look the same, they are the same, so we call that, all right, a bivalent. They're together. Now, if you have a look at, you see this diagram over here, they do something very, very odd. They change genetic material. You don't have a bracelet on or a watch or anything.
No.
Come here quickly, bring your Vaseline. Here, quickly. And your other one, and your other. Come, stand here quickly. I know you're busy. Okay, stand there. Okay, okay. He likes his Vaseline. All right, let's see. Come stand there. Okay, let's have a look. Stand next to me. All right. We are homologous chromosomes. We're the same, by the way. Okay, there's your eye, your nose, your nose. Okay, now what has happened is, all right, now technically, I like Vaseline. And what do you like?
I love the
You love the hand lotion. All right, but these are both creams. Now, I'm Vaseline and he's hand lotion. Right, now when we cross over, what happens? Am I still Vaseline?
No.
I'm mostly Vaseline, but what am I now?
You're hand lotion.
But what are you?
I'm Vaseline.
But what were you mostly Abram, now you're a little bit of Vaseline.
Mm-hmm.
So are we different now?
Yeah.
Yes, we are. Okay, thank you. Okay. All right, that is crossing over. What happened there? Me and Abram were the same. We're two people, okay? But I had a Vaseline and he had a hand lotion. And what we did is we took our things and we swapped them. So in the end, I now am not the same as I started off with, and he's different to what he started off with. That we call crossing over, okay? And crossing over is where all of a sudden, what do? We mix up our genes. We get this variety. And that point where crossing over occurs, we call it a chiasma. Now remember one of the things I said to you about meiosis. What do I want? I want genetic variation. I must mix up my chromosomes. So what have I done here? I have mixed them up. Now I want you, when it comes to homologous chromosomes, this is how often I explain to my kids what a homologous chromosome is. A chromosome looks like this. Okay, now how is it possible to get all these chromosomes into here? Now remember I said I made 46 and I made 46. How do I do that? It's easy. What happens if I make a copy of this? I've got two things. But now I want to keep my copies together. So how do I keep things together? I put a paper clip and I tie them together. So what have I got? This is my centromere, which is my paper clip. And technically speaking, I have 2 chromosomes. But I don't want them to get all mixed up. So I keep them together, all right? And I call that, each of those, because it can't be a whole chromosome, each of those is now called a chromatid. All right? One chromatid plus another chromatid gives me my whole chromosome. Now have a look here. If these 2 pieces of paper, they're actually one, now I made a copy of them. But what have I done? I've used my paper clip to keep them together. So what does it look like? It looks like one, but if I move my paper clip away, which I can do later, I can actually split them. Okay, so the first phase that you look at is prophase. Prophase is a phase they often like to ask. How do you know it's prophase? They're going to show you the chromosomes that are entwined. Okay, they're crossing over. And often they say, what is the benefit of this? And you are going to answer, crossing over enables genetic variation. But now I need to get to the 2 sides. So now what happens to my chromosomes? My chromosomes move to the equator. Now I always remember, if you remember M, M always stands for middle. Okay, where are my chromosomes? They are in the middle of the cell. Do you notice my spindle is nicely attached? Because my spindle is going to pull in. Now, the next thing that happens is very different to mitosis, because in anaphase, if you have a look here, did you notice this is my homologous chromosome? What happened in mitosis? My centromere divided. Here, my centromere doesn't divide. How am I going to get half? If I want to share half a packet of biscuits with Abram, all right, I don't want to break it up and give it to him. We've got a pack of biscuits, all right, and I've got 46 biscuits. And what do? I say, 1 for you, 1 for me. 1 for you, 1 for me, right? And what I do is, on the equator, 1 chromosome goes to the top, because it's Abram, 1 top, 1 bottom. There's no order. Doesn't matter which one. The next one, 1 top, 1 bottom. So we share, right? So what has happened? I have taken that number and I'm starting to get ready for the half. If we have a look over here, how many I had, all right? 1, 2, 3, 4 chromosomes. 1, 2, 3, 4. Now I want to halve the number. So how do I do that? I say 1 set to the one pole, 1 to the other. And it's not a case of you, number 1, need to go. It's random. And that's the next word you need to understand. Random assortment. What does random assortment mean? The chromosomes will randomly move to the equator. So there's no order. And that random assortment ensures genetic variation. Now that my chromosomes are at the poles, telophase occurs. What does that mean? my cytoplasm constricts, my nuclear membrane over here, all right, forms, and technically how many cells have I got? I now have 2 cells. I started off with 4 chromosomes, but I now have got 2 chromosomes in each. So what have I done? I have halved my chromosome number. Reduction, division. Okay. Now, I'm left, all right, I'm left with, say, 2. When we look at the next phase, keep in the back of your mind, you cannot now half the chromosome number again. You can't get 1.5 or a half, right? What do you have to do? You cannot now divide again. So once I've halved my chromosome number, stop. Okay, I've stopped. That's my chromosome number complete. But now the question is, how do I get to the 4 cells? Okay, and that leads us then to our next phase, meiosis II. Now, when it comes to meiosis, how do you know that it's meiosis? First of all, you need to look at what the chromosomes are. Very often, okay, if you have a look, homologous chromosomes are represented like that. At the end, if it looks like half a chromosome, you know it is meiosis II. So let's start. At prophase, prophase 1, I start all over again. There's my IPMAT, but I don't have an interphase. Why do I not have an interphase? Because I do not need to change my chromosome number. I've done that. Okay, now keep in mind my little chromosomes that I have kept all nice and neatly with my paper clip. All right, so now what am I going to do? I need to start the division all over again. So what happens? My centrioles, they're going to start to form. What happens to my nuclear membrane? It disappears. Right? And what do I see again? Look what I see. I see chromosomes. Not homologous. Right? Not homologous chromosomes. Why? Because its partner is in another cell. Here, have a look. There's the one cell. There's the other one. They're no longer together. They are separated. That's what we wanted to do. We want to share the genetic information. So now one set is in the one cell, one is on the other. Now I have got 2 cells. Now please keep this in mind. I do not want to divide my number again. I want to keep it the same. So how do I do that? I line up on the equator, middle. There we go. Do you see the middle? I line up on the equator. There we go. I'm lined up. And you see how nicely I'm lined up? You know why I'm not, sorry, let me just quickly do that. Okay. You know why I'm so nicely lined up in the equator? Because what I want to do now is I want to take my paper clips and I want to take them off. So in other words, what do I do? What did I say? This is technically actually 2. So it lines up on the equator, and imagine it's a zip. Imagine it's a zip, because if I unzip, I'm going to cut everything down the center. If it's not in the center, if one is here and one is over here, it's going to make it very difficult for me to try and separate. So why do I put it in the middle? It's easy, because it gets ready for what I want to do next. And what do I do next? I separate the centromeres. So I had this. I take the centromere and I divide it in half. I take my paperclip out and I free my 2 chromosomes. You see what happens here? I don't change the chromosome number. What I do is I break them apart, and the centrioles are going to pull again. And when those centrioles pull again, what do? They pull them to the other sides of the pole. And when they pull them to the other sides of the pole, I've got one set of information here, one here. Because remember, it's one cell that again divides in two. One cell that divides in two. So at the end, when I want to share all my cytoplasm, how many cells do I have? 1, 2, 3, 4. They all have half the chromosome number, and they all look different. Are we ready for a break?
Yes.
I think so, hey? All right. Okay, and
Nice
then I
one.
can come back to the next thing, and we can start the questions.
Looking forward to it, hey?
Yes.
And I must say a big shout out to everybody that is watching the show with their moms. This is so special. I've got three guys that are watching. Lerato Matising, enjoying the show with my mom. She likes the show because she also learns a lot. She is a life science teacher. A big shout out to you, Mom. Mpumi Gulepel say, I'm watching the show with my mom. She is helping me in the number of chromosomes. Proud of her, all thanks to Mindset. And lastly, Batabile Matenti saying, my mom didn't know all of this, but sure, she is learning a lot, and so do I. Thank you, Mindset. Thank you, guys. We'll see you after this break. Don't go. Welcome back, guys. This is your segment, guys. It's all about you and your questions and your comments. But remember, we've got the Beer Bright Spark competition. It's closing on the 3rd of July, and the winner will be announced live on the 21st of July. So enter right now. Who is taking home the next Chevy Spark L? It could be you, and I could be visiting you saying, congratulations! So guys, enter right now, and more details are on our Facebook page. Remember, be a Bright Spark and enter to win right now. Now, Cheryl, I've got this one simple comment for you. You know, before you continue.
Before I continue.
I thought I was gonna give you during the break, but hey,
Okay.
here's a surprise for you. This is from Emmanuel Maluk saying, please shout out my name, Emmanuel.
Emmanuel.
You guys are really effective. Can you please ask Ma'am Cheryl, what must we expect in the paper that we are writing on Wednesday?
I'm not, not coming to me. Ask him what he's writing on. Maybe we can see.
Yeah, if you've got questions that you'd like Cheryl to help you on, we'll help you guys. Remember, guys, we don't give you guys, a specific scope, but everything that we're doing, it is a scope. So take whatever that we are learning as your scope. We're not going to give you a question paper. We don't
know what.
Yeah, we don't know either.
Yeah.
So take it
All
over,
right.
Cheryl.
Good luck for your test, Emmanuel. I hope you do well. I don't know what you're writing on. My biggest thing is please do past papers, do practice questions. You don't focus on the type of the question, but it gives you an idea of what they're going to ask you. Right? I'm going to do little questions after this. I just want to do the last thing. The last thing what you need to look at meiosis is when something goes wrong. All right? So what during meiosis, remember I said to you, all right, what needs to happen? We have these stages where things separate, or I move one to this side, one to the other side. Sometimes that doesn't happen. Say I'm sharing my cookies with Abram again, and I go one to you, one to me, one to me. One to you, one to me. What I do is I skip him out, and I get double, right, and he gets one less. And that is going to have an influence if that egg, all right, is fertilized. So when we talk about, so now we look at the numbers. As soon as I say one for you, one for me, one for you, one for me, one for me, all of a sudden he's going to have one less. And that could have an effect on if the egg is fertilized. Or so an egg could be -1, an egg could be +1. We call that when the separation doesn't occur properly. In other words, instead of my paperclip not dividing, my centromere not dividing, one whole one going, or when I'm supposed to go one for you, right, I take an extra one. A very important word, please. You need to know it. It's called non-disjunction. And quite simply, it means that separation doesn't occur when it should. Very important. Know that word. Now you're going to link it to your genetics. Why? Have a look here. You've done the Down syndrome before. And record trisomy because what number are we supposed to have here? We should only have 2. Now, because of that, what occurred in meiosis, I didn't share the number of biscuits with Abram properly, which means is I got an extra one when I shouldn't. And that extra one has an influence, all right, on what is going to happen. There's two that I've also looked at. You don't have to know, but you very often, please make sure you link Down syndrome with non-disjunction. There are two other syndromes that you just briefly, you don't need to know all the disorders. Have a look here. In males, some males have got an extra X. What has happened? My sharing with Abram the biscuits, I have kept one for myself, and it will have, all right, bring about certain features over there. For the female as well, Turner syndrome. What is Turner? And those of you who watch NCIS, I put a picture of Hetty over there because she actually has Turner syndrome. She has got one less. So she would be Abram's thing, where I stole one of her biscuits and she has one less than she should, which can bring about some kind of complications. Okay, let's start with the questions. Remember what I say to you? First thing you do is you have a look. What you think of. Number one, I need to think, which phase is it? Oopsie, sorry. Which phase? Why is it that phase? And number two, can I label all my different phases? Okay, so let's have a look here. Without starting here, what do we notice? Okay, if I have a look at my diagram, when I look over here, I'm already starting to think, look what's happened, all right? Do I see the characteristic homologous chromosomes? No, I don't. So if I don't see homologous chromosomes, maybe I'm looking at phase number 2. What I've also noticed is that crossing over has occurred. So again, it gives me some idea, right, that what is happening, that this phase is occurring in meiosis II. But let's see if they ask me that. Now here we go. Question number 1. State which phase of meiosis is represented in the diagram above. Now, what you notice now is they're not in the middle. There's my IPMAT. Okay, so crossing over has occurred, can't be prophase. M stands for middle, can't be metaphase. After they're in the middle, they get pulled to the poles. What is happening to them? They're getting pulled to the poles. So my phase is anaphase. But now, which one? Which one is it? Now, if I have a look over here, what can I see happening? They said chromatid. So basically, what is a chromatid? It's a half. I have separated. When do my chromosomes separate? During the second phase. Okay? So I am then, my phase is anaphase 2, not anaphase 1. Now it asks me, all right, so just to make sure that you didn't have a lucky guess, they're going to say to you, so, okay, so why? What's the reason? And there's your reasoning. Let's have a look. Okay, there's a few things. Number 1, no homologous pairs. Number 2, chromatids, right, have separated. And once they are separated, that's only. What's separated? Actually, the centromeres separated. And where does the centromeres occur? Only in meiosis II. And why do I know that it's an anaphase? Because there I'm telling you why it's in the meiosis. Why is it in anaphase? Because I know in anaphase, my chromatids, always in anaphase, where do they go? Are pulled to the poles. Okay, so have a look for your mark allocation. They might be asking you one reason why it is anaphase, and for your second mark, why did you say it was in number 2 and not number 1. Okay, now we need to be able to identify. Let's label. I see there's an A and I see there's a B. Number A, that would be my easy one. Let's mark it. That will be my spindle fiber. Right, now number B, lots of you get confused with this and you tell me it's the nuclear membrane. It's not. What happens during meiosis? The nuclear membrane disappears. So number B is my cell membrane. You very often think that this happens in the nucleus. No, all right? It happens in the cell. My chromosomes were in the nucleus in the beginning, and then what did I do with them? The nucleus is in the way. Get out of the way. Disappear. Let me do my thing. Right? So my A obviously was my spindle. Please write there A so they know. And B is my cell membrane. Be nice and neat and orderly. Okay, now it says here, how many chromosomes were present in the parent cell before it underwent meiosis? Okay. What do we know about meiosis II? Meiosis II, I have half my number of chromosomes. Okay? Now you guys must be very careful. How many did the parent have at the beginning? If I divide this, I am going to get one cell with 4 and one cell with 4, correct? Because it's going to divide. So now, how many did I have in the beginning? My parent cell would have been, all right, if I had half here, so I had 4 and I had 4, what would my parent cell have had? They would have had 8. Okay? They would have 8 chromosomes. Now it says, will be present in each cell at the end of meiosis. Look at my cell. Here we go. Let's make another line. There's my one cell. There's my other cell. And I only always have 2 choices. Half of the original, but here it shows me 1, 2, 3, 4. There we go. 4 cells at the end. Remember, what is the whole thing about meiosis? Reduction, division. That's how much I started off with, 2n. I need to halve it, n. Okay, the last one. State one place in the body of a human female. Right, it's very specific, the question. One place in the body of the human female, and where do we make it? You guys should know the diagram because you have done reproduction, and in a female, meiosis is called oogenesis, which makes an egg cell. And where do we make the egg cell? In the ovary. All right, so it's specific. They ask where in the female. If it was in the male, then we would say testes. Could the cell represented in the diagram be that of a human? All right, and your question is quite simply no. And why do I say no? Look at the number. All right, explain your answer to question 1.6. Humans have, how many do we have? We do not have 8 chromosomes. This you need to know. Humans have 46 chromosomes. So the cell, if human, should have had 23. All right, not 4. We do not have 4. That's why it cannot give it. All right. Now this is the one thing you need to know all the time. Please, definitely, please. Why is meiosis biologically important? Biologically important means why must it happen? What are the 2 things that I've told you? What do we need to do? So, we need genetic variation. It ensures we get that. How did we do that? I'm going to write here just in case they haven't asked you. Crossing over and random assortment. All right, and what else do we need to get? Genetic variation. We need to half the chromosome number. Why else is it important? Okay, to maintain species chromosome number. All right, I would go for, when it comes to the first, I would usually go for the first two. Halving the number of chromosomes, right, and genetic variation. As you can see, right, the questions are very much what we say phase orientated. Here's the next one. Study the diagram below and answer the questions that follow. As soon as you see that, what do you notice? It's the first thing. As soon as you see that, you should immediately crossing over. All right, crossing over. You need to be able to identify crossing over. Here it goes. First question even says to us, name the process occurring in the diagram. Look there. Right, it's showing you, me and Abram with our Vaseline and our thing. What did we do? We crossed over to each other and we changed. Right, so that is called crossing over. So nice, often meiosis can be quite an easy question. Right, provide a label for the region marked X. What is X marks the spot where two meet? It's called a chiasma. Okay, now, during which phase of meiosis does this process occur? These are things you need to learn. These are things you need to know. Prophase. And if you leave out the number, it is wrong. Where does it occur? Number 1. Where do I get all my genetic? All right, all my differences in genetic material? In number 1. And the last question, have a look again. The same question as in the previous one. List one important process, all right, importance of this process and why. Now be very careful here. This, you could get this of this process is not meiosis. What is the importance of this crossing over? Because you might say it halves the number. It doesn't. The question is relevant to the process at the top of the page. Look at your diagram. They're asking you that process. And what is the importance of that? Crossing over is genetic variation. It's not halving the number. Meiosis 1, yes, but not the particular process at the top. How are we doing for time? Yeah, we're almost there. Yep. All right. Okay, I'm going to, guys, just have a look. Go through it. You see, all the questions are very much phase related. Is that it?
Yeah,
or do you need more time to spin your spin? Are you fine?
That's it.
Okay.
Thank you so much,
It's a pleasure.
It's been a great show. I really enjoyed it. And so the guys at home, I must say, Busisi where Precious is having a birthday today.
Oh,
so
happy birthday.
This
Wow.
was the best present ever.
Sure.
And all the best to everybody that is writing this week or next week. We wish you nothing else but the best. Always make sure that you have this workbook, guys. It's only 139 rands, guys. For more details, ask me and I'll tell you on Facebook. And we appreciate you guys. But a big thank you to the Department of Basic Education for making sure that we have this show. Without them, it wouldn't be possible. We love you guys and we always believe in you guys. Have a good evening. Peace.